Welcome to AP Biology. We're in the cell signaling unit, and I want to talk about receptors today. So that's the first step in the signal transduction pathway. The first thing is that a ligand has to attach to a receptor, and some of them are actually embedded in the cell membrane, most of them actually. And so what we're going to do right now is talk about how these three examples of receptors are the same and how they're different. So one thing that's the same, this is a G protein coupled receptor, tyrosine kinase receptor, and a ligand-gated ion channel. So how are they the same? Well, they all have a part of them that's on the outside here, right? So this part is outside the cell. And then they all have a part that actually spans the plasma membrane, right? Like this. And then they all have a part of them that's on the inside, facing the cytoplasm here. And so we're going to see some similarities just based on that. So we're going to look at the similarities and then we're going to look at the differences. One thing to notice first is that water-soluble or hydrophilic ligands are going to bind to receptor proteins that are in the plasma membrane. If it's a hydrophobic or not water-soluble ligand, that could go right through the lipid bilayer, right? Because things like testosterone, they're lipids, they're hydrophobic, they mix right in with this hydrophobic lipid bilayer and they can go right through the cell membrane. But other molecules, the water-soluble ones, the hydrophilic ones like insulin, they can't go through the lipid bilayer. And so they're going to typically be received by some receptor that has a portion of them that's outside here. And so let's look at these in general. A protein's domain is a region of a protein's polypeptide chain that's self-stabilizing and folds independently from the rest. So we're going to use this term protein domain. So here's a protein. This is a tyrosine receptor tyrosine kinase, which we'll talk about in a little bit. But what is the same about all the different receptors on the cell surface? Well, they have a part of them that's on the outside. And so the domain means the part of the protein that's folding and ends up out here. We're going to say this is the ligand binding domain of the receptor protein. The ligand would bind here and here, but we're going to call this entire thing the ligand binding domain because it's the part of the protein that's folded and ends up outside of the cell. And then we're going to say that this part right here is the intramembranous domain of the receptor protein. So it's the part of the protein that's actually in the cell membrane itself. And so remember, the cell membrane has hydrophilic parts to it, right? But most of the cell membrane, the inner part of the cell membrane, that's hydrophobic. Those fatty acid tails repel water. And so what you need to remember with this is that the protein itself here, remember the proteins are made out of amino acids, right? A protein is made out of a bunch of amino acids bonded together. But do you remember that amino acids have R groups attached to them? So the amino acid goes nitrogen, carbon, carbon with an O, right? But they have this R group. So this would be the amino acid. And the R group can be hydrophobic or hydrophilic or polar or whatever, or neutral, I guess. But this R group can be hydrophobic. And so the R groups of the intramembranous domain, they can be whatever as they fold and are in the center. But the ones that are on the surface of this part of the protein are all going to be hydrophobic because they're mixing in with the lipids all around them. So I don't mean the surface like outside the cell. I mean the surface of this part of the protein itself has to snuggle up next to the lipids here. So that part has to be hydrophobic as well. That part of the protein has to be hydrophobic. And then there's the intracellular domain, the part that's inside the cell. That's this part. A lot of times they're going to call this the cytoplasmic domain. It's the part of the protein that's actually facing the cell, that's inside the cell. And that part has some part of it that does something. Often it's an enzyme. Sometimes there's a tube that lets something through. But you're going to need to be able to determine the differences between the different parts. So hydrophobic in here and probably hydrophilic out here and here, because remember, outside the cell there's water and inside the cell there's water. Okay, so let's look at each individually. So there's a G protein coupled receptor. there's the tyrosine kinase, and then there's the ion channel receptors. So let's look at this first one. Let's talk about the G protein coupled receptor. So here's what it looks like, and it has the outside, the part that ends up on the outside here. This is outside the cell. And these, remember, a protein is made out of amino acids bonded together. And remember how the order of amino acids is the primary structure? And then it forms these loops called an alpha helix. And so there's an alpha helix right here. That's the secondary structure. Then it loops around and does another. So it goes through and loops a whole bunch of times. So this G protein coupled receptor will loop back and forth like that. I don't know, 2, 4, 6, 8. I think they always go in and out with eight of these loops, which isn't super important. But notice that right here, the R group here would be hydrophobic, and here, and here, and here, and actually the other surfaces too, all the way around, right? So this would be the intramembranous part or intramembranous domain, and then the parts out here are the cytoplasmic domains. So maybe this is the segment that interacts with a G protein. I haven't even told you what a G protein is yet, so let's talk about that. So this whole receptor here, we're going to call this a G protein coupled receptor. Okay, it's a receptor, great. It can accept a ligand. Okay, super. Coupled means like, oh, how sweet. Right? They're a couple. Coupled means it works with something else. So it gets together with this thing called a G protein. So a G protein coupled receptor is a receptor that works with or couples with a G protein. Okay, great. What's a G protein? Right? It works with a G protein. A G protein acts as an on-off switch. There are a whole bunch of different types of G proteins. And they bind to GTP or GDP. If they're bonded to GDP, it's inactive. If it's bonded to GTP, it's active. So let me show you that. You might remember ATP because we spent a whole lot of time on it during the cellular respiration and the photosynthesis unit, but it is actually an RNA monomer. It's a nucleotide. So a nucleotide is made out of a sugar and a phosphate and a base. And you might even remember that RNA nucleotides come in 4 different forms, A, U, C, and G. And so this one is adenine. It's this one. And it happens to actually have 3 phosphate groups, which is what the TP comes from. So adenine refers to this, and the TP is the triphosphate. It's got 3 phosphates. And remember, if that last phosphate comes off, now it's not 3 phosphates, it's not triphosphate, it would be adenine diphosphate, right? Okay, so ATP isn't the only interesting molecule. GTP is another molecule that we're going to talk about, and it's important in this unit. So the difference here is that it's got a guanine base instead of an adenine base. But it also can have the 3 Ps, right? And so that makes it guanine triphosphate. If it loses that last phosphate, we're going to call it guanine diphosphate. So here is what it looks like. Here's the receptor. The ligand would fit right here, but it's not there right now. So right now it's inactive. This is the G protein. So a G protein is a protein that can interact with GDP or GTP. If it's got the diphosphate, it's inactive. And then here's some enzyme that's going to be involved in a little bit. They each have a part of them actually that's in the lipid part of the cell membrane. Okay, so here's a signaling molecule that attaches to its particular receptor. Remember, these are very specific connection, right? So if this is insulin, the receptor is only for insulin. Once this signaling molecule attaches to the receptor, there's a change in shape. We're going to call that a conformational change. It's just a shape change. When that happens, this G protein can now attach, and when it does, the GDP leaves, and GTP, the active one, can come on, and now this is a G protein that's activated. Once it's activated, it's going to hop over here to this enzyme and activate the enzyme. And that enzyme can activate some other enzyme. So maybe it says, maybe this is insulin, and it's saying, hey, enzyme, activate some reaction that's going to pull glucose into our cells, that's going to open up a transport protein and pull glucose in. And then it eventually gets inactivated. Remember, these molecules can all bond on and bond off, right? Hop on, hop off. So they're not going to bind on and attach forever, right? It's a I can't think of the word I want, but they kind of hop on and off. So it's reversible. That's the word I wanted. They can come on and off. So here's the summary of that. Here's the G protein receptor with no ligand. So everything here is inactive. The GDP is what's showing you that it's inactive. Once the signaling molecule, the ligand, attaches to the receptor, now it changes the shape in here of the receptor. This G protein attaches to the receptor, and then GDP leaves and GTP hops on. Now it's activated. It can come over here and activate this enzyme, which then causes some kind of response. And this is reversible, so it all hops off and the whole thing gets inactivated again. So that is one type of membrane protein. That's a receptor.